So, good afternoon, everyone. Today, we'll be starting with the discussion week 11 of the biochemistry course of NPTEL. Okay, so in this course, we have studied about the beta oxidation of fatty acids and the synthesis of the fatty acids in mitochondria. Okay, so let's start. So, this is the first question for today. Which of these are false regarding the oxidation of fatty acids? So, what can be the correct answer? So the correct answer is option number B, C, and D. So let's go through the options. So the saturated fatty acids undergo beta oxidation. Yes, both saturated and unsaturated fatty acids undergo beta oxidation. So this is correct. Then option B, the fatty acids are broken down in the mitochondria one carbon at a time. No, this is false. Okay, so when beta oxidation takes place, so two carbons are removed from one fatty acid molecule. Okay, so this two carbon compound is the acetyl coenzyme A that actually enters the TCA cycle. Okay, so these are two carbon compounds that are derived from the mitochondria, derived from the fatty acid rather than one carbon at a time. Okay, so this option is false. Omega 3 fatty acids means a double bond that exists between the chain's second and third carbon starting from the methyl group. Okay, so considering this as the fatty acid, for example, so this fatty acid is actually a 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbon uh, fatty acid that have a double bond at ninth position and 12th position. So there are two forms of fatty acid labeling. First is that starts from the carboxyl group of the fatty acid. Okay, so that is the general nomenclature that we use for labeling the fatty acid. And the second one is starting from the omega carbon. That is the last carbon from the of the fatty acid. That is the methyl carbon that we say. Okay, so the omega nomenclature starts from the methyl group and from where the double bond starts. Okay, so the first double bond is at the sixth carbon from the methyl group. Okay, so that means this is an omega 6 fatty acid, and in case of omega 3, the third carbon from the methyl group would have the double bond. So this is the omega 3 fatty acid. Okay, so the option says that omega 3 fatty acid means the double bond exists between the chain second and third carbon starting from the methyl group. So no, that means the double bond exists between the third and the fourth carbon. Okay, so this is false. Then an omega 3 fatty acid means a double bond that exists between the chain's third and fourth carbon starting from the carbonyl group. No. So omega 3 fatty acid, as I said, the nomenclature starts from the omega carbon. Okay, so this option is also false. Sorry. Only fatty acid chains containing more than 12 carbons require shuttle system their transport across mitochondrial membrane. Yes, this is correct, because the more than 12 carbon fatty acids actually are bulky. Okay, so they cannot transport across the pores that are present on the mitochondrial outer membrane. So that's why they require a shuttle system to get it transported through the mitochondrial membrane. So this is correct. So the false options are option B, C, and D. Okay. So any doubts in this question? Okay. Let's move on to the next question. Identify the correct statements from the following. So these are the statements regarding the beta oxidation of fatty acid. So what can be the correct answer? Okay. So the correct answer is B, C, and D. Okay. So this is the normal beta oxidation of palmitoyl coenzyme, that is palmitoic acid. Okay. So let's see the cycle first, then we'll see the options. So palmitic acid has 16 carbons. Okay, so now the actual oxidation starts from the first two carbons, that is the starting from the carbonyl carbons. Okay, so the first step is dehydrogenation, in which the coenzyme A molecule is actually dehydrogenated with the help of FAD molecule. So one molecule of FADH2 is removed here. Okay, and this leads to the formation of a rearrangement or the double bond at this position. Okay, so this forms trans delta square delta 2 enoyl coenzyme A. So that this delta 2 actually means that a double bond is inserted at the delta 2 position. Okay, so that means at the second position. So this is the second carbon. So a second position, the double bond is incorporated. Okay, now a water molecule is added and enoyl form is actually being formed. Okay, so this is called as enoyl coenzyme hydratase enzyme, and it actually inserts a water molecule and it dissolves the double bond that was formed earlier. Okay, now the compound that is being formed is beta hydroxy acyl coenzyme, because here a OH molecule is actually added in the compound. Now the other dehydrogenation reaction takes place with the formation of NADH. Okay, and here the enzyme that acts as beta hydroxyacyl coenzyme dehydrogenase. Okay, now the compound that is formed is beta ketoacyl coenzyme. So here a keto group is added at the third carbon of the compound. So why? Because this compound is actually being broken off from the fatty acid chain as a acetyl coenzyme. So this is the two carbon compound that is being uh, extracted from a fatty acid molecule. So to extract that, this keto group is introduced at the third carbon. So now After extracting a acetyl coenzyme A molecule, the acyl coenzyme A acyl transferase, that is thiolase, actually forms a two carbon less compound. Okay, so that means initially the fatty acid is C16. So after removal of an acetyl coenzyme A, C14 carbon, that is acyl coenzyme A, or the C14 or mystryl coenzyme A is actually formed. Okay, now this cycle continues further, the same thing for another like 6 cycles to form the acetyl coenzyme A. Okay, and this acetyl coenzyme A actually enters the TCA cycle. Okay, so now let's see the options. So the conversion of palmitoyl coenzyme A to trans delta 2 enoyl coenzyme A requires enoyl coenzyme A hydratase? No. Okay, so this is actually a dehydrogenation reaction. So this is the first reaction of the beta oxidation. So this requires a dehydrogenase. Okay, so the first option is false. Then conversion of L-beta hydroxyacyl coenzyme A to beta ketoacyl coenzyme A requires beta hydroxyacyl coenzyme dehydrogenase. Okay, so yes, this is correct. So from beta hydroxyacyl coenzyme to beta ketoacyl coenzyme, it requires beta hydroxyacyl coenzyme dehydrogenase. So this is correct. Then in beta oxidation of palmitoyl coenzyme A, NAD plus is converted to NADH and H plus. Yes, this is also correct. Okay, at the third step, this reaction takes place. So this is also correct. The final product of beta oxidation of an odd numbered fatty acid molecule is propionyl coenzyme A and acetyl coenzyme A. Yes, this is correct. Okay, why? Because if there is a odd number carbon, then the final product will be a 3 carbon compound and a 2 carbon compound. So propionyl is a 3 carbon compound and acetyl coenzyme is a 2 carbon compound. Okay, so this is also correct. So the correct options are B, C, and D. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. Which of the following enzymes participate in the beta oxidation of acyl chains with even numbered carbons? So what can be the correct answer? So the correct answer is option C and option D. Okay. So so acyl coenzyme acyl transferase actually takes place in the carnitine pathway. Okay, to transfer of fatty acid to the mitochondria. And G-enoyl coenzyme hydratase actually takes place here in the beta oxidation. Okay, so this happens to be the transport enzyme, and this happens to be the involved in beta oxidation. Okay, so these two enzymes are actually involved in the oxidation of acyl chains. Okay, so let's move on to the next question. Which of the following coenzymes or cofactors function in one or more fatty acid oxidation pathways? So, what can be the correct answer? Okay, so the correct answer is actually A, B, C, D. Okay, so this is NAD molecule that is involved in the beta oxidation. Biotin is involved in synthesis. Okay, as well as the it is involved in yes, synthesis, and then cobalamin is also involved in oxidation. So this is vitamin B12, and acetyl coenzyme A is the final product of the beta oxidation. So this is also involved here. So both A, B, C, D are the vitamins that are involved in the oxidation or synthesis of the fatty acid part. Okay. Okay, so let's move on to the next question. Which of the following are true about the regulation of fatty acid metabolism? So, what can be the correct answer? B, C, and D. Okay. So the correct options are B and E. Okay. So let's see. Okay. So this we have discussed in the last class also. Okay. So now we'll be discussing about the uh, pathway. So that is the molecular pathway. So the dietary carbohydrate actually increases the blood glucose, that leads to increase in the production of insulin. This actually increases phosphatase activity. Okay, so this leads to the uh, inactivation, or so yes, so the activation of the ACC protein. Okay, so ACC is a protein that actually converts acetyl coenzyme A to melanoyl coenzyme A. Okay, so this melanoyl coenzyme A leads to the production of fatty acids. Now, this melanoyl coenzyme A allosterically inhibits carnitine acyl transferase 1. Okay, so that actually converts fatty acyl coenzyme A to fatty acyl carnitine for the transport of the fatty acids to the mitochondria. Okay, so now in the mitochondria, the beta oxidation takes place. Now, contrary to this, when the blood glucose levels are low, that means glucagon levels are high, and this activates protein kinase A. Okay, so this phosphorylates ACC proteins and inactivates it. So that means acetyl coenzyme A is not converted to melanoyl coenzyme A. Okay, so if melanoyl coenzyme A is decreased actually, so that means now the fatty acid acyl coenzyme is actually converted to fatty acyl carnitine, and it gets moved to the mitochondria for ATP production, because here the blood glucose levels are low. Okay, so now the ATP production goes back to the fatty acid pathway. So let's see the options. Melanyl coenzyme A stimulates carnitine mediated transport of fatty acyl chains. No, it inhibits. Okay. Then acetyl coenzyme A carboxylase is regulated by the phosphorylation and dephosphorylation. Yes, this is correct. Okay, so this ACC is acetyl coenzyme A carboxylase enzyme. Low blood glucose levels stimulate acetyl coenzyme carboxylase activity. No. Okay, it inhibits because it activates protein kinase A, it phosphorylates ACC, and that actually cannot convert acetyl coenzyme to melanoyl coenzyme. So that it inhibits. Okay. Fatty acyl chains in the mitochondria transported to the cytosol to synthesize membrane lipids. No, this is false. Okay, because fatty acyl chains once enter the mitochondria, they get they actually enters the beta oxidation pathway, so they cannot be transported back to the cytosol. Phosphorylation of perilipin makes the lipid droplets accessible to lipase. So this was the lipase wala pathway, and here after activation of perilipin, it actually activates it. Yes, it makes the lipid droplets accessible to the lipase. Okay, so this is also correct. So the correct options are B and E. Okay, rest of the options are false. So any doubts in this question? Okay, so let's move on to the next question. Which of the following inhibit as fatty acid beta oxidation? So what can be the correct answer? B and C. Okay, so the correct answer is A and D. Rather than A, it would be B. Okay, because phosphorylated uh, acetyl coenzyme carboxylase actually inhibits the formation of sorry, it would be A and D only. Okay, so high blood pressure actually leads to inhibition of the fatty acid beta oxidation because it has to dilate its the blood vessels. Okay, so that actually needs space. So that's why the beta oxidation is actually inhibited at that time. Then phosphorylated acetyl coenzyme carboxylate, it increases beta oxidation because it leads to decrease in the level of melanin coenzyme. Okay, so it leads to the, increase in the transport of fatty acids to the mitochondria. Okay, then phosphorylated perilipin. So phosphorylated perilipin actually leads to increase breakdown of lipid droplets. Okay, so these also lead to increase in the beta oxidation. Then high levels of melanyl coenzyme A. So if high levels of melanyl coenzyme A, this would inhibit the transport of fatty acid to the mitochondria. So this would inhibit the this would inhibit beta oxidation. Okay, so the correct answer is option A and option B. Okay, any doubts in this question? Okay, if not, then we'll move on to the next question. Which of the following statements are true regarding the biosynthesis of fatty acids? So, what can be the correct answer? So the correct answer is A, B, C, and D. So let's see the options. So the source of CO2 for biotin carboxylation is the bicarbonate ion. Yes, this is correct, as we can see in the image also. So this is a biotin arm of the biotin carrier protein. And first, the bicarbonate ion is added to this, and this is done by the biotin carboxylase enzyme with the help of ATP. Okay. Now, this carbon dioxide is added, then trans carboxylase enzyme actually adds a acetyl coenzyme group to this. So this forms the malonyl coenzyme A. Okay, so now this malonyl coenzyme A actually so this is how the reaction takes place. So this helps in the formation of the malonyl coenzyme A. So that helps in the formation of the fatty acids. Okay. So let's see the option. So the first option is correct. Then carboxylation of biotin is ATP dependent. Yes, this is also correct. So this is the first step. Carboxylation of biotin is catalyzed by the enzyme acetyl coenzyme A carboxylase. Yes. Uh, no, actually, it is catalyzed by the enzyme biotin carboxylase. So this is false. Okay. Trans carboxylase enzyme is responsible for the formation of malonyl coenzyme A. Yes, this is correct. The formation of malonyl coenzyme A is a reversible reaction. No, this is false. So the correct options are A, B, and D. Okay, not C. It's A, B, and D. Okay. So any doubts in this question? Okay. If not, then we'll move to the second next question. Which of the following is incorrect about the fatty acid biosynthesis? So what can be correct answer? Okay, so the correct answer is B and C. So, in the first step of the fatty acid synthesis, what happens? This is the one SH group where the acetyl group is attached. Okay, and here is the milanoyl group is attached. Now, this is the fatty acid synthase enzyme. So, first step is condensation, where the CO2 molecule is released and leads to the formation of this 4 carbon compound. Okay, then the reduction happens where this double bond is actually reduced to, and add H and OH are added here. Then dehydration for release of this H2O molecule to transfer this double bond to the second carbon. Okay, then again reduction happens so that there is no double bond in between, and this is the 4 carbon uh, saturated acyl chain. And here, the after the melanoyl group, two carbons are added. So likewise, two carbons are added after each other from the acetyl coenzyme A. Okay, so let's see the options. CO2 is released during condensation of the acyl chain and melanoyl coenzyme A. Yes, this is correct. So this condensation in CO2 is released. The product of condensation reaction is a 5 carbon acyl chain. No, it's a 4 carbon chain. Okay, because one carbon is released in CO2. Then CO2 is released from a 5 carbon product to yield a 4 carbon acyl chain. So a 5 carbon compound is not at all formed. Okay, it is rearranged, and here this CO2 group is actually released. Okay, so a 5 carbon compound is actually not formed. So this is also false. Then fatty acyl chain are extended by 2 carbon units in each cycle. Yes, this is correct because of the acetyl coenzyme A. The addition of carbons to the chain is a four-step process. Yes, this is also correct. First is condensation, then reduction, then dehydration, and then again reduction. Okay, so this is also correct. So the incorrect options are B and C. Okay, so any doubts in this question? Okay, so let's move on to the next one. Which of the following enzymes takes part in the fatty acid biosynthesis? So what can be the correct answer? Okay, A, D, and E. Okay, so the correct answer is A, B, C. And so for fatty acid synthesis, all the enzymes that are involved are part of a single ACP, that is acyl carrier protein. And the different enzymes are beta ketoacyl ACP synthase, then acetyl coenzyme ACP transacetylase, enoyl ACP reductase, beta hydroxyacyl ACP dehydratase, beta ketoacyl ACP reductase, and then melanoyl coenzyme ACP transferase. So these are the six enzymes that are part of a a single complex, and so that means they have six different active sites. Okay. Now let's see the options. We have melanyl coenzyme A ACP transferase. Okay, so yes, we have melanyl coenzyme A ACP transferase. This is correct. ketoacyl ACP reductase. We have beta ketoacyl ACP reductase, correct. Enoyl ACP reductase, yes, we have enoyl ACP reductase. Then we have beta ketoacyl synthase, no, ACP synthase. Okay. Then butyryl ACP reductase, no. Then we have trans delta 2 butanoyl ACP dehydratase, no. So these three enzymes are not there. So ABC is the correct answer. Okay. So any doubts in this question? Okay. So let's move on to the next question. Which statements about the acyl carrier protein are incorrect? So what can be the correct answer? Okay. So the correct answer is option B and C. So these statements are incorrect. So this is a acyl carrier protein that have a serine side chain. Okay. So this is the serine side chain that is attached to the pantothenic acid, that is vitamin B5. Okay. So this is the whole molecule is 4-phosphopentothenate. Okay. And then this SH group is the sulfhydryl group that is esterified to the malonyl groups. Okay, so this is the whole part of the acyl carrier protein. So let's see the options. So it has vitamin B5 derived coenzyme. Yes, this is correct. So pentothenate is the vitamin B5. It has an alcohol group where the acyl chain binds. No, it has a sulfhydryl group where the acyl chain binds. It catalyzes the conversion to the of the malign group, malonyl group to hydride. No, this is also false. The incoming malonyl group binds to the sulfhydryl group. Yes, this is correct. So the incorrect options are B and C. Okay. So any doubts in this question? Okay, let's move on to the next question. Which of the following statements are incorrect? D, E, F. Okay, so the correct answer is A. That is incorrect. Acetyl coenzyme exits the mitochondria as malate. No, this is false. Okay, the conversion of malate to pyruvate by malic enzyme requires NADP plus reduction to NADPH. Yes, this is correct. So malic enzyme actually converts malate directly to pyruvate. Okay, so this requires vitamin, that is NADP, that is reduced to NADPH. Okay, then citrate positively regulates the activity of acetyl coenzyme carboxylase. Yes, this is correct. So this citrate actually regulates the activity of acetyl coenzyme carboxylase and this too positively. Okay, so this is correct. Then palmitoyl coenzyme inhibits acetyl coenzyme carboxylase. So this inhibits this enzyme, palmitoyl coenzyme A actually inhibits this acetyl coenzyme carboxylase. So this leads to inhibition of conversion of acetyl coenzyme to melanoyl coenzyme. Okay, so this is correct. Then insulin positively regulates citrate lyase activity. Yes, this actually regulates the citrate lyase activity by converting citrate to acetyl coenzyme. So this is also correct. Acetyl coenzyme carboxylase is inactivated by phosphorylation. Yes, this is also correct, by the activity of glucagon, epinephrine. So they activate protein kinase A that actually phosphorylates acetyl coenzyme carboxylase. Okay, so that is actually inhibited by phosphorylation. Okay, so option A is only incorrect. The rest of the options are correct. Okay, so any doubts in this question? Okay, so let's move on to the next question. Which of the following are true about the beta oxidation of fatty acids? B and C. Okay, let's see. So the correct answer is B and D. Okay. So the for fatty acyl chains longer than 12 carbons, the oxidation happens with enzymes in the mitochondrial matrix. No. For any fatty acyl chains, the oxidation obviously happens in the mitochondrial matrix. Okay, so this is false. The conversion of beta hydroxy acyl coenzyme to beta ketoacyl coenzyme is similar to conversion of malate to oxaloacetate in the TCA cycle. Yes, this is correct. So these are the three pathways, three steps in the TCA cycle. Okay, conversion of succinate to fumarate, then fumarate to malate, and malate to oxaloacetate. Okay, so these three steps are actually similar to what is happening in the beta oxidation. Okay, so palmitoyl coenzyme converted to trans delta 2 enoyl coenzyme, then L beta hydroxyacyl coenzyme, and then beta ketoacyl coenzyme. Okay, so now beta hydroxyacyl coenzyme conversion to beta ketoacyl coenzyme is a dehydrogenation reaction. Okay, so this is equivalent to malate to oxaloacetate conversion in where the malate dehydrogenase takes enzyme acts on it. Okay, so this is correct. Then enoyl coenzyme hydratase can hydrate the naturally occurring double bonds in fatty acids. No, this is false. Okay, then beta oxidation of myestoyl coenzyme followed by TCA cycle and electron transport chain finally produces 94 ATP. Yes, this is correct. So, uh, first of all, we have to check what is the carbon number in the myestoyl coenzyme. So, this is actually 16 carbon. Sorry, it's 14 carbon. Okay, so it's 14 carbon. So that means it will take uh, 6 cycles to convert this 14 carbon to 7 acetyl coenzymes. Okay, so from one myestoyl coenzyme, we'll get 6 NADH molecules, 6 FADH molecules, and 7 acetyl coenzyme. Okay, so after this, what will happen? It will go to TCA cycle, then this will go to the ETC directly. So if this enters the TCA cycle, it will yield one acetyl coenzyme will yield 10 molecules of ATP. Okay, so directly 7 acetyl coenzyme would yield 70 molecules of ATP. Okay, so one NADH molecule actually yields 3 ATP molecule. Okay, so this would be 18 ATP. Then from one FADH, it yields. Uh, Wait a minute. Yes, yes. So one NADH molecule when enters ETC, it yields 2.5 ATP. Okay, as per the recent convention, and one FADH2 molecule yields 1.5 ATP. Okay, so 6 NADH means it will yield 15 ATP. Okay, and 6 FADH means it will yield 9 ATP. So if we total these three, it comes out to be 94 ATP. Okay, So the fourth option is also correct. So the correct options are B and D. Okay. Any doubts in this question? Okay, so let's move on to the next question. Patients with vitamin B12 deficiency have been found to have propionate and methyl malonate in large amounts in their urine. What could be the possible reason? So, what can be the correct answer for this? Okay, so the correct answer is option B. So actually, vitamin B12 is required for the enzyme that converts the methylmalonyl coenzyme A to the product. Okay, so if it's not present, that means the enzyme that is actually making that is converting methylmalonyl coenzyme A stops. Okay, so that leads to accumulation of methylmalonyl coenzyme A, and due to this accumulation, high amounts of propionate and the methylmalonate are present in the urine of the vitamin B12 deficiency patients. Okay, so any doubts in this question? So let's move on to the next question. When malonyl coenzyme A produced from a labeled 13CO2 and unlabeled acetyl coenzyme A is used as in the biosynthesis of palmitic acids, where is the final product will be labeled carbon be present? So what can be the correct answer? Okay. So the correct answer is option C. That means the labeled carbon would be absent in the palmitic acid. Okay, and here is the explanation. So when malonyl coenzyme A is formed, this carbon dioxide is the first group that is added to the acetyl coenzyme A. The first step of the reaction is condensation that releases this carbon dioxide. Okay, so that means if this is labeled, this would be released as carbon dioxide, and the acetyl coenzyme is actually added to the acetyl coenzyme of the malonyl group. Okay, so no labeled carbon would be present in the malonyl group after the condensation reaction. Okay, so that means no labeled carbon will be present in the palmitic acid. Okay, so any doubts in this question? Okay, so let's move on to the next question. Which of the following statements are true? So the okay, A and C. So correct answer is option A only. And in beta oxidation of 11 carbon saturated fatty acid, the direct products are 4 acetyl coenzyme and 1 propionyl coenzyme A. So let's see, we have a 11 carbon saturated fatty acid. So acetyl coenzyme A has 2 carbons, so this will be 8 carbon, and propionyl carbon has 3 carbons, so this totals to be 11 carbon. So yes, this is correct. Then we have 10 cycles are required for the complete oxidation of arachidonic acid, that is a 20 carbon saturated fatty acid. So for a 20 carbon fatty acid, it would require 9 cycles. Okay, so this is false. Then malonyl coenzyme ACP transferase transfers group from ACP to the cysteine of the beta ketoacyl ACP synthase. No, this is false. Insulin, glucagon, and epinephrine, all three of them negatively regulate fatty acid biosynthesis. No, they actually positively regulate biosynthesis by activating the enzyme acetyl coenzyme A carboxylase. Okay, so they actually positively regulate acetyl coenzyme carboxylase. So this option is also false. Okay, so the correct answer is option A only. So any doubts in this question? Okay, so let's move on to the next question. When carnitine acyltransferase 1 is mutated such that its functionally capabilities are unaffected, but it can no longer recognize melanoid coenzyme as substrate, which of the following is true? So what can be the correct answer? Okay, so A and B. Okay, so let's see the answer. It's A and D. So, as we know, malonyl coenzyme A negatively regulates the carnitine acyltransferase 1. So, that means fatty acyl coenzyme A will not be able to convert fatty acyl carnitine, and it will not be able to transport to mitochondria. Okay, so that means malonyl coenzyme A is not being like recognized by the acyl uh, uh, carnitine acyl transferase 1. So fatty acid will be converted to fatty acyl carnitine and it will be transported to the mitochondria. Okay, so the first thing will be that it will not be able to inhibit the fatty acid oxidation, and then this leads to the fatty acid oxidation and biosynthesis actually will be unregulated. Okay, because this is the major pathway where the fatty acids are actually being transported to the mitochondria for beta oxidation. So if this pathway is already not regulated, that means it will be a continuous cycle of oxidation and fatty acid biosynthesis. Okay, so let's see the option. Melanoid coenzyme can no longer inhibit fatty acid entry into mitochondria. Yes, this is correct. Beta oxidation comes to a halt. No, beta oxidation will never halt in this case. There will be no effect. No, results in futile cycles of fatty acid biosynthesis and oxidation. Yes, this is correct. Okay, because now transport is not regulated at all. Okay, so that's why this will not be able to restrict the beta oxidation at all. So options A and D are correct. Okay. So any doubts in this question? Okay, so let's move on to the next question. Each cycle of beta oxidation produces the following. So what can be the correct answer? Yes. So one cycle of beta oxidation releases one NADH, one molecule of FADH2, and one acetyl coenzyme. Okay, so option D is correct. Okay, so this is the beta oxidation pathway. So this one FADH2 is released, one NADH is released, and the acetyl coenzyme, one molecule each. Now let's move on to the next question. How many cycles of beta oxidation are required to fully convert the molecule shown below into acetyl coenzymes? So what can be the correct answer? Okay, so let's first count the number of carbon atoms that it has. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Okay, so irrespective of the presence or absence of double bond, okay, the number of cycles of beta oxidation would remain the same. Okay, so it will only depend upon the number of carbon atoms the fatty acid have. So it has 18 carbon atoms. That means if each cycle would release one acetyl coenzyme A, that is a 2 carbon compound. So the number of cycles to produce the acetyl coenzyme A's would be the number of carbon atoms by 2 minus 1. Okay, so this is a general formula that you can use to deduce this. So number of carbon atoms was 18 by 2 minus 1. So this would be 8. Okay. So any doubts in this question? Okay. So this is the end of the today's presentation. So if you have any doubt regarding any concept of this speech, you can ask, or any questions in this presentation. Okay, if not, then we'll close the session for today. Okay, thank you for joining in. Thank you.